Hello and welcome to lecture M6.25, Introduction to Bioinformatics. The learning outcomes for this module are as follows: to provide an introduction to bioinformatics, an introduction to the Human Genome Project, and to explain some applications of bioinformatics. There are other videos associated with this screencast which are in the lecture folder. First, very short videos, under 2-3 minutes. There's a general introduction to bioinformatics, and then there are two other videos on the Human Genome Project sequencing, the public and private projects. So these are under 2 minutes and under 1 minute indeed. So from each of these, what I'd like you to take away the issues with repeat sequences in there and shotgun sequencing. Just two terms to watch out for in those videos. They're very straightforward, gentle introductions to what's going on. So a definition for what bioinformatics is. And I think firstly what I'd like to start with is this is an absolutely enormous field in its own right. And all I'm attempting to do here is to give students a broad introduction. So, what is bioinformatics? The application of computer science and information technology in the field of biology and medicine, and that is fairly straightforward. I think you probably could have made that definition up yourself. So, where this becomes important is we're using information systems generally to analyze large biological data sets, so huge amounts of information being analyzed by computer, huge amount of information generated from biological observations. And an example of that you may be familiar with is DNA and protein sequences, and we'll look at those a little bit in more detail in a second. So as I say, it's a broad term and it can relate to these subdivisions as well. So it has a whole study of its own right in algorithms, developing and applying algorithms, ways to manipulate and interpret data. So not only do we have to get the data, we have to know what to do with it and how to change it or transform it into a format that we wish to understand or attempt to understand. But curiously then, maybe you may not link the two, databases and information systems such as those available in the National Library of Medicine in the United States, and I've spoken about them before. And then developing software, mining data, going back into large data sets and literally digging out important information, hence data mining, as in coal mining or diamond mining. Modeling and simulation, which we'll see a bit more of in a minute, and basic statistic analysis or complex statistic analysis. So very mathematical and very computer based, but all applied to biological data. There are two broad goals in bioinformatics, and these are important definitions: to uncover the wealth of biological information hidden in the mass of data. So we're trying to dig in and find out what's going on, and also to obtain a clearer insight into the fundamental biology of organisms using information. Now this is and will have profound impacts on the provision of healthcare in the future. It is that unlocking, if you like, analysis and unlocking of the huge, vast amounts of information that we currently have some idea of, but as computational power increases and indeed analytical methods increases, this will become much more relevant and give us a clearer insight into what's going on at a fundamental level. So if we just take, and I'm sorry to preach to the converted here to a degree, it's just I want to do basic biochemistry here. If we take, and it's just because the next few slides contains some terminology, I want everybody to be with me on this. If we take a central point that DNA makes RNA, makes protein, that will be basic biochemistry. And the sequence of the proteins determine its structure, how it's actually physically put together, and its structure determines its function. Some other basic terms, and these are important. The genome is the complete set of genes in an organism, hence the Human Genome Project, which is one of the videos, or two of the videos I want you to watch. Genomics is the study of genomes. Proteins are very simply, you know, complex molecules, chains of amino acids. Proteome, the entire complement of proteins in a cell. And proteomics, the study of protein structure and behavior. And this maybe is the most important definition on the page, these two, genomics and proteomics. So, why is this important? Well, a major task in computational molecular biology, informatics, bioinformatics, is to decipher information contained in biological sequences. So if we can chop something up and look at its constituent part, what does that mean? And if we're looking at nucleotide sequences in a genome, this is all the information necessary to produce the functional organism. We looked at all of those information. We should be, in theory, able to figure out what's going on. And we could decode it and perhaps duplicate this decoding using computer systems. So if we can get to that level of detail and then reassemble it, we can understand what's going on. So some basic applications in bioinformatics would be the mapping and analysis of DNA and protein sequences. The aligning of different DNA and protein sequences, compare them, so from two samples, and creating and viewing 3D models of protein structures to analyze or predict their function. So in a lot of cases, proteins, drugs interact or are modeled in 3D theoretically before they're actually developed in the lab. Now from that, and I just mention again, there's two videos there under 3 minutes in total, the public and private part of the Human Genome Project, and all students should watch and understand what's going on in those two videos. So let's talk about the Human Genome Project as a very good example of bioinformatics in action. So this is really an international scientific research project, so that initially it was a global event, if you like, and basically put to determine the sequence of chemical base pairs which make up DNA, Human Genome Project. And it was set up to identify and map approximately 20 to 25,000 genes of the human genome from both a physical and a functional standpoint. We want to know what these genes were doing. And it began in 1990. And 10 years later, there was a working draft of the human genome announced. 10 years work in many, many locations around the world. And it was declared completed in 2003, still 10 years ago from now. And it is still further being analysed, published and refined. So we're looking at 23 years of work to date to map the human genome. So this is a mammoth task, and this little graphic would show you everything printed on a shelf here, about 35,000 genes in the human body. And of these, there's 40,000 bases. The gene varies in size, as you know. And still at this stage, the functions are unknown for over half of the discovered genes. So we're now looking at the stage trying to figure out the function of the genetic code. So the average human gene produces 3 different proteins, so the complexity is immense. But the work continues. And just to give you a better look at that, that would be all of the chromosomes. You see them all labelled there. That is filling an entire bookcase, and they're tiny, tiny writing on each page. You get the idea. It's a huge amount of information. So applying that to bioinformatics, we can analyze the sequence information in that and predict 3-dimensional structures. And this then can be visualized, giving researchers more the experimental results. They can see the results more clearly than just by looking at raw numbers. So it gives us two ways to look at data. And if we take a look here, there is a sequence of base pairs. No idea where it's from, but you can see an awful lot of information. If it means something to you, well done to you. But when that is actually visualized then and put together, we get something that looks like this, which is entirely different. We can look at functional groups and bonding sites on this molecule, giving more and more information. So going from here to here obviously has great advantages. So why is this important? Why should we bother with all this information? Why bioinformatics? Well, what we're doing is making sense, as I said at the start, of that wealth of data that has been produced by these sequencing projects. We've got a lot of raw data, and now we have to operationalize it and make a difference, make some inferences, make some discoveries. So the sequence is generated, it has to be stored, it has to be, there's an interpretation, and there's an analysis, and none of this can happen without computer software and computer hardware. So the challenges specifically for the Human Genome Project is massive amounts of information which has to be stored intelligently. We have to be able to get access to it and retrieve it and look after it and put it back together, and indeed, if we discover something, return it to databases so it can be used by others. And that's where it has to be easily and reliably accessed. So the data itself, although there's lots of it, it's absolutely meaningless, and this is really important unless we analyze it. And unless we've written the algorithms to make that analysis, the data will remain meaningless. So again, the data doesn't produce the result, it's how we interpret that result. So we're continually developing these tools to extract meaningful biological information. Now, there is an ethical issue to this, and this is quite important. And I have mentioned in a previous lecture about HIPAA, H-I-P-P-A. It's an act of government in the United States. Well, let me get to that in a minute. The previous time I mentioned it, it was about privacy, and it's similar here. But the Genome Project raised a considerable amount of fears amongst people in that increased knowledge of the human genome could be used to discriminate against people. And the discrimination could be based on their genetic profile, obviously, meaning if people were more disposed to disease or genetic conditions, they might find it difficult to get jobs or to get health insurance. And this became such an important point that HIPAA, the Health Insurance Portability and Accountability Act, was drafted into law, and it protects against the unauthorized and non-consensual release of individually identifiable health information to any entity not actively engaged in the provision of healthcare services to patients. Now, that's exactly the same as its relevancy in the previous lecture, where we talked about privacy generally. But here you can see why it would be so important. If somebody were to have access to my genetic information, and they will say, well, we're not going to insure this man because he has this disorder. Or if we think a little into the future, it may be the case that we're full sure he's going to get some type of disease when he's 60. So there is a fantastic film called Gattaca, which covers this with Jude Law. If you ever get a hold of it, watch it. The whole world is run by a genetic profile. A little bit far-fetched for the moment, but certainly not within the realms of possibility as we move into this huge area. So don't forget that with this increased knowledge, there are vulnerabilities. Somebody's genetic profile, it's a powerful piece of information, but in more ways than one. So some other examples of application of the field of bioinformatics, and I'll quickly give you these. I'm going to look at molecular medicine, genetic diseases and gene therapy, and antibiotic resistance. But there are many, many more examples. Just let's give you 3 of them here. So let's have a look at this molecular medicine. So this is a disease linked to genetics, and estimated there could be 4,000 hereditary diseases on the human genome. So, for example, in the Irish context, quite a lot of higher incidence of cystic fibrosis in this country, a lot to do with the genetic makeup of the country. So we could have a disease linked to genetics, we could have something that happens that alters the genome from an environmental factor or stress, such radiation causing cancer, or indeed diet and heart disease, or diabetes and diet. So an analysis of the human genome can find genes directly associated with different genes. And if we were to understand the molecular basis of these diseases, we may be enabling better treatments, cures, and possibly preventative tests. And that's by getting down to the level of understanding the specific gene. And this has already happened in many, many cases, that people are screened for specific diseases. One springs to mind is a breast cancer gene, which when expressed, the usual radical surgery is a double mastectomy, such as Angelina Jolie opted for after discovering the gene from a genetic test. So gene therapy in genetic diseases, the potential for using genes themselves to treat a disease. So this is where genes could be manipulated directly in the patient, and then changing how that patient's genes behave. So it's actually directly manipulating the genes within the patient. So that many diseases run in families, for example, and that would be again the BRCA, I think is the name of the gene that Angelina Jolie was tested for. So these run in families and a result of genes which predispose family members to illness. Some examples here: Alzheimer's, cystic fibrosis, breast cancer, colon cancer, heart diseases, and that again, the BRCA, I believe, is the name of the gene there. And one single gene in cases of cystic fibrosis, which is very interesting, can be called the Celtic gene, getting back to my earlier point about it being very prevalent in Ireland. And the complexity there, that heart disease, there could be 20 or 30 genes involved that actually come together and coalesce to give the heart disease, to form the heart disease, but less and less understood than cystic fibrosis. So this, just before I go on a little bit, this Richard Resnick, this is an optional video. You may wish to watch it, but he talks about genomic revolution, and I think it's quite an amazing talk. It's only 11 minutes. You might enjoy it, but it's not directly relevant to the course. And then if we look at antibiotic resistance, we can examine the genome of bacteria that cause infection in hospitals. And say hospital patients is a huge issue all around the world, hospital acquired infection. And we can look up certainly what we're looking for, and what's been found is what they call a pathogenicity island, or region made up of a number of antibiotic genes that may contribute, antibiotic resistant genes that may contribute to the bacterium's action. So what researchers are trying to find is, why are antibiotic resistant bacteria, why are they resistant? So we could mark these up and try to establish controls that spread the infection in wards. So we can get in, and if we can understand what's going on in the bacteria, we may be able to stop the spread of the infection. So this is, to conclude, this is a huge subject, and I hope I've given you some insight, not obviously the whole thing. There are many organizations. Probably the biggest one, but better funded, is the European Bioinformatics Institute. But there is no doubt, as you're starting on your career, this will have a significant impact on the provision of healthcare in the future. But don't forget that also bioinformatics can be databases, information that is readily available. And you should maybe go and take a look at a few of these. And I mean, we're back in PubMed, those databases. Here's we can query the human genome. There's a GenBank. There's lots and lots. You can start here. It's very easy to find. But we'll look at the Entree Global Query Cross Database Search System. So what that is allows users to search many discrete health sciences databases for biotechnology information. And this gives us views of genes and protein sequencing, chromosome maps, all that kind of thing. So the information has been produced, it's been made sense of, it's been tagged, and it's been made available to the public free of charge. So try and watch the other videos in the folder as well, the Human Genome Project, and thank you very much for listening.